Selamat siang. Halo?
Halo.
Iya,
Siang, Prof.
oke. Saya ini kan terima email dari
Oke.
siapa ya, pokoknya dari panitia untuk
Oke.
klik itu aja. Nah, sudah saya klik, kok nama saya jadi keluarnya Takashi Matsumoto ya? Oh, karena saya nggak
Sampun
ada.
kami rename, Prof.
Kenapa?
Sampun kami rename.
Oh, ya udah, oke.
Ngapunten, Prof.
Selamat siang. Ini nanti kita mulai open buat attendee-nya jam 2 ya. Jadi opening ceremony-nya jam 2. Untuk semua jam 2 kita mulai. Good afternoon, Professor Ludovic. Thank you very much for joining us today. We will start our workshop at 2 p.m. in Indonesian time, and in London time it will be 6:40. Okay, no, 7 o'clock a.m. from in London time. So please wait a while. Thank you very much.
Thank you.
Professor Ludovic, today you will have time for presentation. Professor Ludovic, you will have time for presentation for about 30 minutes. Is it okay?
30 minutes? Okay,
Yeah, 30 minutes. And then after that,
Okay.
we continue to panel discussion.
Okay, so 30 minutes. And just to confirm the time, so the time will be 3:20 local time.
In Indonesia, it will, for your presentation, it will be at 3:20 p.m. in Indonesian time.
Yeah.
And in London time,
Okay.
it's 8:20.
8:20. And that's for 30 minutes. Okay, great. Fantastic. Okay. Cool.
30 minutes, yeah.
Okay, thank you very much.
Okay.
Because on the program I received, it was an hour, but I'm happy to shorten my talk to 30 minutes.
Oh, yeah. Okay, thank you very much.
Thank you.
Bukannya 1 hour ya?
Hello, Professor Ludovic. I'm sorry for my mistaken information. So for your presentation time is about 1 hour from 8:20 until 9:20, but you have
Okay.
to prepare before your presentation about 30 minutes before your presentation time.
Okay, good. Okay. I will be around anyway. Okay, that's good.
Okay, okay, thank you very
Okay.
much.
Is there any time for question for the presentation? Is it no?
Yes, after your presentation, there will be a discussion time.
Okay,
So,
okay.
yeah, so all the participants will like drop their question in
Join
the
the-
Q&A box, and then you can like choose which question will you answer.
Okay, good. Great. Thank you.
Okay, thank you very much. Okay, let's start our opening. Good afternoon, ladies and gentlemen. Welcome to our international workshop series. First of all, allow me to extend my warmest welcome for you all to this international workshop series. Gastroenterology research opportunities in antimicrobial resistance, metagenomic, clinical surveillance, and wet lab experiment. Hosted in Surabaya, Helicobacter pylori and Microbiota Study Group, Institute of Tropical Disease, Universitas Erlangga, has the honor to present you this workshop. I would like to introduce myself. My name is Rizky Amalia, and I will be the master of ceremony for today's workshop. Recently, due to global pandemic, most of all universities, offices, and other public places are being temporarily shut down. However, for students, lecturers, and other academicians, they continue their education through online learning and studying from home in order to improve their knowledge. With this online workshop, the excellent speakers will share their up-to-date knowledge regarding gastroenterology research opportunities in antimicrobial resistance, metagenomic, clinical surveillance, and wet lab experiments. Before we open our international workshop together, let's sing Indonesian national anthem. Ladies and gentlemen, to mark the commencement of this international workshop series, we are very honored to have the welcome remarks delivered by Dr. Langgen Waskito, MD, PhD from Institute of Tropical Disease, Universitas Erlangga, Indonesia, and also Professor Maria Inga Lucida, MD, PhD, Head of Institute of Tropical Disease, Surabaya. I am now very pleased to invite Dr. Langgen Agung Waskito, MD, PhD, to deliver his welcome remark. Dr. Langgen, the time is yours.
Okay, thank you very much, Ms. Rizky Amalia, for very exciting upcoming workshop today. So this workshop series is actually our commitment for accelerating every single researcher and colleagues in the Universitas Erlangga and also other university to increase their ability, their insight, their idea for very good research in the future. That's why in this upcoming workshop, we are inviting several speakers from 6 countries. They're coming from Japan, Taiwan, England, and also from United States. We hope that with their insight, a very powerful insight from those international speakers could give us a lot of idea and technique that we can apply to our upcoming research. So yeah, actually we are expecting that our potential participant will be around 100 and 200 participants, but now it's already registered participants about 300 and something. So I hope that this is going to be very, very exciting discussion, and also a lot of knowledge that we can share and we can gather for everyone who join these upcoming workshops. Okay, so welcome for everyone, and thank you very much that you are joining this workshop. This is our really our fuel to give you the best thing that we can deliver to all of you, and hope you can gather a lot of insight and a lot of more technique that you can apply for your upcoming research. Thank you very much, and welcome to our very, very exciting workshop series.
Okay, thank you very much, Dr. Langdon, for your impressive speech. Now, I'm pleased to invite Professor Maria Inga Lucida, MD, PhD, to deliver her welcome remark. Professor Inga, the time is yours.
Yeah, thank you, Ms. Rizky. Before I speak, there are 62 participants not coming yet, still in the waiting room or something. I saw here participants 63, but yeah.
Maybe they don't attend in time, like maybe a little bit late, because we have like the time is until 3. Yeah, this workshop will be held until 5:20 p.m. in Indonesian. So maybe the participant, we don't know in which time they will join us.
Okay. Honorable Professor Ludovic Baye, MD, PhD of University of Cambridge, UK. Pardon my pronunciation. Professor Yoshio Yamaoka, MD, PhD of Oita University, Japan. Professor David Graham, MD, PhD of Baylor College of Medicine, Houston, Texas, USA. Professor Ratakon Vilayjone, MD, PhD, FACG, AGAM of Thammasat University Hospital, Paduntani, Thailand. Pardon my pronunciation. Dr. Wei Chi Liao, MD, MSc, PhD, Associate Professor of National Taiwan University College of Medicine, Taipei, Taiwan. Dr. Takashi Matsumoto, PhD of Oita University, Japan. Dr. Pavini Subsomwong, PhD of Hirosaki, Aomori, Japan. Pardon my pronunciation. Dr. Muhammad Miftah Surur, MD, PhD of Erlangga University. My colleague, Dr. Langgang Agung Waskito, MD, PhD. Dr. Dala Dohan, MD, PhD. Also to Dr. Judith Anissa Ayu Reskita, Dr. Firman Setiawan, Dr. Kartika Afrida Vausia, Dr. Astri Dewayani, Dr. Raisa Firki Trianda, and Dr. Ricky Indra Alvarai, professors, seniors, doctors, and all participants. Good afternoon, good morning, good evening to all of you. Assalamualaikum warahmatullahi wabarakatuh. It is undoubtable that university rankings do matter, and research citations are among several important performance indicators being assessed, which strongly related with scientific paper performance. Universitas Erlangga, which is one of the prominent universities in Indonesia, is enthusiastic to intensify its position in the university ranking. Institute of Tropical Disease, Universitas Airlangga were recognized and awarded by former named Indonesian Ministry of Research, Technology and Higher Education as a Center of Excellence in Tropical and Infectious Diseases in Indonesia since 2012. As a COE for higher education, ITD Universitas Airlangga is dedicated to integrated and advanced medical and health sciences, of which various graduate fellows could join, such as medical doctors, dentists, pharmacists, veterinarians, biochemists, microbiologists, chemists, physics, biologists, and many others. Some of our outstanding prestigious international research projects were JGRADE, Japan Initiative for Global Research Network projects in infectious diseases, already 3 terms with 5 years for each term. And then SATRAPS, Science and Technology Research Partnership for Sustainable Development Project on medicinal plants against hepatitis C virus, year 2010, year 2014, and again SATRAPS on searching lead compounds of anti-malaria and anti-amoebal agents, year 2015, 2020. There are 16 study groups in ITD, and in this opportunity, Helicobacter pylori and microbiota study group holds an international workshop titled Gastroenterology Research Opportunities in Antimicrobial Resistance, Metagenomic, Clinical Surveillance, and Wet Lab Experiment. We are very grateful to several prominent and reputable speakers from several countries, UK, US, Taiwan, Japan, Thailand, also our prominent senior scientists, as well as our young talented staffs of Airlangga University for having been sharing their expertise in this workshop. I do hope and I am sure that participants would get some insights and benefits on the new trends, up-to-date information, and opportunity in gastroenterology researches from this workshop. Thank you. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you very much, Professor Inge, for your very impressive speech. Now, ladies and gentlemen, I would like to proceed our main agenda for today, for the International Workshop Series Day 1. The first honorable speaker for today is Dr. Dalla Dohan, MD, PhD from Institute of Tropical Disease, Universitas Airlangga, Indonesia. Before we start, let me introduce Dr. Dalla Dohan's profile. This is Dr. Dalla Dohan profile. You can see in the screen. Okay, now I have the pleasure to invite Dr. Dohan to give the presentation about the best practices for polymorphism study. Dr. Dohan, the time is yours.
Thank you for the moderator. Can you hear my voice?
Yes.
Okay, clearly, okay?
Okay.
Okay. Good afternoon. First, let me introduce myself. Can I share my screen immediately?
Yes, of course, you can share your
All right.
screen. Excuse me.
Sorry, how I can start my screen sharing? Okay, can you see the presentations right now?
Yeah, can clear.
Okay, pretty clearly. Okay. Good afternoon. First, let me introduce myself. My name is Daladuhan, and I came from Institute of Tropical Disease, Universitas Erlangga, Indonesia. And first of all, I want to give my highest respect to Helicobacter pylori and microbiota group for successfully held such a great event. And thank you to give me such a good opportunity to share my knowledge. I hope that this event will be going well from the beginning until the end. So today I got the opportunity to share my knowledge about single nucleotide polymorphism detections using PCR RFLP methods, the applications in the labs, and association with gastroduodenal disease pathogenesis. So for the introductions, let us talk about gene polymorphisms. So gene in our human genome, gene sections in which the base pair of the sequence is the same for all the DNA are called monomorphic, while the other sections in which the sequence of nucleotides different or more than one alleles occupies the gene locus are said to be polymorphic. Uh, there are countless places in the human genome where actually differ between peoples. In this picture, in the left picture here, I give you illustrations of the variations between normal, carrier, and also with disease. So in normal individual, for example, here in the chromosomes and the bulk of the chromosome, for example, here it's with G or guanine, and then complement with cytosine, and it applies in both chromosomes. While in the carrier, in one of the chromosomes here, change from G into A or adenine and complement with T or thymine. And in disease here, in both chromosomes, because human have is a diploid, so we have two chromosomes. So both G change into adenine in both chromosomes. So this is just a picture just to illustrate one types of polymorphisms. And in the other picture also give illustration that in many locations in the chromosomes between individual, we have a lot of variations. For example, here in individual 1 might be different if we compare to individual 2, individual 3, individual 4, or even individual 5, and so on. And this kind of variations occur very frequently in our human genome. So approximately around one mutations every 1,000 letters code. So it means maybe in human genomes, we have approximately around 3 million variations, this kind of variations. And it raised a question for us. So what's the difference between polymorphism and also mutations? So the alleles at the polymorphic sections are called either mutants or common polymorphisms, depending on their frequency on the populations. So expert says that if it's less than 1% in the populations, we call it mutations, while if it's more than 1%, range between 1% until 6% or 7%, we can call it polymorphisms. However, sometimes it's pretty difficult for us to distinguish between this common polymorphism and also in mutations in our populations, especially if there is no data about the polymorphism before in that populations. Okay. For the types of polymorphisms, polymorphism itself can occur in protein form or also in DNA form. So for the protein form, the most common examples, the easiest examples is ABO blood groups, where we have variations of people with A blood group, B blood group, O blood group, or AB blood group. While there are several various types of DNA polymorphisms, the most common one is the single nucleotide polymorphism, SNP, or we can pronounce it as SNPs. So like in the picture here, in the upper picture, This is one example of the SNP. So it characterized by only the chains of only one nucleotides in the genome. And it usually the most common types of polymorphism here in the human body. And then the second type is tandem repeat polymorphism. So tandem repeat polymorphism is a condition where short length of DNA that are repeated multiple times within a gene. So for example, in the second picture here, we can see that there is a repeat unit of CAG, and then it's in the chromosome 1 different with the copy 1 different with the copy 2, where in the copy 2 the CAG repeat unit is repeated a couple of times more than the copy 1. And then also another common type of polymorphism is small scale deletions or insertions. So insertions is when the DNA is inserted into the new place, while the deletions is when the nucleotides is removed from the genome. For the classifications of SNP, okay, now we talk deeper about SNP, single nucleotide polymorphisms. So based on the, we can categorize the SNP based on several types. Based on the chains of nucleotides, we know about transition and transversions. So transitions is when purine nucleotides change into another purine, or pyrimidine change into another pyrimidine. So in this case, purine is, for example, is adenine and guanine, and pyrimidine is cytosine and thiamine. While transversion is the change from the purine nucleotides into pyrimidine nucleotides, or vice versa. Based on the expressions, polymorphism also can occur in non-coding region or coding regions. If it's in coding regions, it can be expressed in synonymous or not alter the amino acid. It means even though the amino acid is changed, the amino acid it expressed, it's still the same. But if it's non-synonymous, after the polymorphism change one nucleotides, it affects the amino acid and then it will express as another amino acids. And based on the allele pairs type, we know that about homozygous and also heterozygous. And then how we detect the SNP? So currently there are various methods we can use to detect SNP, and the most one of the oldest method, and then the most common method we usually use is enzyme-based methods, or we call it, for example, is polymerase chain reactions restriction fragment length polymorphism, or PCR RFLP. We can pronounce PCR reflip to make it easier. So this method is the one that we will talk deeper in these sections. And then another method is PCR followed by direct sequencing, and then also hybridization methods using probe, and then SNP analysis using next generation sequencing. Okay, this picture shows, give us illustrations about two types of methods to detect SNP. One is using direct sequencing. So in the upper picture, we can see section A, B, C. In the section B, the arrow showing to T, it means the peak here shows that the sequence read is thiamine. And in the C, picture here, it's read as C. So it means in the human genome here, in the B is genotype is TT, homozygous. And in C, it's a CC, homozygous also. But in picture A here, we can see that we have two peaks here. These two peaks, from the color, we can distinguish that this is peaks of T and C. But even though this the sequence say that it is T, however, we have two peaks here. It means this is heterozygous. This is not homozygous. It means the genotype is CT. This is one method using direct sequencing. Another method is using hybridization method. In this case, we use a special probe. here, this probe contain specific nucleotides complement to the polymorphism here. So for example, in case this is a perfect match to the polymorphism, it will attach and start the extension process. And then after the extension process starts, the DNA polymerase will active and then release. The D1 here is the dye, the one that will give the signal. And then after it's being released, it will give a signal that we can detect using some equipment. And then conversely, if for example in the probes, the nucleotides here, it's not a complement to the polymorphism, it will not attach, and then even the DNA polymerase will not working, and then the dye here, D2, will not release, and then we will not get any signal at all. That's how we are able to detect using hybridization method. And then why we have to use PCR reflip? What's the advantage or what's the disadvantage? Of course, its method have its own advantage or disadvantage. However, for PCR reflip, we have several advantages compared to the others. First, high reliability. So because it is generated from the specific sites via non-restriction enzyme, the results are constant over time and the locations. And then the second, using this method, we are able to distinguish between heterozygotes and also homozygotes. And the third, high sensitivity and specificity. And then the fourth, it's relatively easy to perform and interpret the result. And fifth, sequence specific, and the result of this method is reproducible. It means even though we repeat the experiment in different time or different places, as long as we use similar protocol, the result will be similar. However, of course, this method also have the disadvantage. For example, we have to perform multi-steps and also time-consuming, of course. And then the biggest disadvantage is this method can only check specific mutations at the enzyme cut sites, which limits the identification of whole genome sequence variations, because we can only check one site at one time. Okay, now that now we move into the application in the laboratory. So now we are going to talk about the technical applications in the labs. This is the workflow of the PCR reflip experiment. So basically, we have three main processes. First, in the left side here is DNA preparations, and then followed by gene amplifications, and then followed by enzyme digestions. So in this discussions, we will talk in about every single step of this PCR reflip experiments. Okay, part one, we will talk about the DNA preparations. So in this step, we have three steps we have to follow, from the sample acquisitions, followed by DNA extractions, and also followed by DNA check. For the sample collections, so basically from human, we can obtain DNA from almost all of body parts. However, the most common samples for DNA extraction can be obtained from blood, and then buccal mucus also possible, and then hair and nail also possible, and also surgical or biopsy specimens. It's important to note that the red blood cell, we are not able to get the DNA from red blood cell, because as we know that red blood cell doesn't have the nucleus, and then also lacks the nuclear DNA and also another organelle. So even though we try to extract the DNA, we will not get any DNA from the red blood cells. Okay, for the DNA extraction here, so currently there are a lot of methods actually for the we can use for DNA extractions. The one of the oldest method, and then even until now we are still commonly use this method, is phenylchloroform methods, and the other is commercial extraction kit. So for the phenylchloroform method is the oldest DNA extractions, and using this method The advantage is we are able to collect almost all the DNA from the specimen. So it is a great option if we need to collect DNA in high quantity. However, the disadvantage of using this phenylchloroform method is that it takes longer time to perform the experiments. It also has a lot of multiple steps. And ultimately, we know that phenol is a dangerous substance. So we have to handle it with extra careful in safety bands, also facility with good circulations. While for DNA extraction using commercial kit is very convenient. It is very only contains maybe very simple step and then relatively easy to use, constant and stable result. And but the disadvantage, of course, the price compared to phenylchloroform method, it's relatively more expensive. However, basically, both methods using similar principles for DNA extractions. So the principles still contain a lysis method, a lysis step, precipitation, and also purifications. Okay, after we successfully extract the DNA, before we start continue the next step, it is important for us to check the DNA we are extracted. There are two parameters important for us to check. First is the concentration, and then the second is purity. So we can check the concentration and purity using spectrophotometer. And for purity, this is very important. For the purity, we check the purity by using the ratio of the absorbance at the optical density 260 and optical density 280. If the ratio is less than 1.8, it means that our extracted protein got protein contaminations. If the ratio is between 1.8 until 2, it means we get the good purified DNA. And if the ratio is more than 2, it means we have RNA contamination here. This graph also shows us that the graph of the good DNA. So the peak, the highest peak here should be 260, and 280 slightly lower than 260. Okay, for the part 2 is gene amplifications. So in this step, we are going to amplify a specific genes, our target, by using PCR or polymerase chain reactions. And then we have to check the result of the PCR by using gel electrophoresis. So as we know that amplifications using PCR, the aim is to obtain enough amount of DNA, because in the next step, in the enzyme digestions, it is mandatory for us to obtain good amount of DNA. So that's why we perform this experiment, PCR, so we are able to get enough amount of DNA. Briefly, maybe I will tell you about the principles of the PCR. So basically, there are 4 principal steps of PCR. First is denaturations. Here, the denaturations is the step when we try to open the double-strand DNA into single-strand DNA. Usually, we use temperature more than 90 degrees Celsius here. And then the second step is annealing. So annealing is the step where the primer is start to anneal to the single-strand DNA. This step marks the beginning of the DNA synthesis. So this is mandatory. And then the next step is elongation process. This is when the DNA synthesis by the DNA polymerase is starting. And then final elongations. And then start again from the first step, second step, third step, and then repeatedly until we have a good amount of DNA. For PCR reflip itself, I have some tips for everyone who will do. So it is better to use primer set that is able to produce PCR products with long enough size. With I advise between 400 until 900. because a short PCR product size may result in even shorter DNA band after digestion, and it will make it harder for us for interpretations. Maybe I will give you the picture in the later slide. This is also another illustrations about PCR. So basically from the double strand DNA here, from the denaturation spa, it will become a single strand DNA. And then in the annealing process, the primers will attach to this single strand DNA and marks the beginning of the DNA synthesis. And then elongation process becomes 2 copies, and then repeatedly again denature, anneal, and then elongation again until we repeat it into several cycles, and then we get millions and millions of copies of DNA. Okay, this is gel electrophoresis to ensure that we successfully amplify the target gene. This is very important because if we are not able to amplify the gene, we are not able to continue to the next step. For this process, we can use either agarose gel or also polyacrylamide gel. And I also have some tips for you here. So for the next step, after we perform this gel electrophoresis, make sure that the DNA concentration of the product is good enough. We can check the concentration by the density here. If the density is very low like this, it means the concentration is low. But if the density is high, the concentration is also high. This is very important because in the next step, if we use this kind of low concentrations, low density, it will make harder for us when we interpret the band after the digesting process. Okay, now we move to the part 3. This is enzyme digestions. So in this step, we will perform DNA digestion using restriction enzyme. And then after we perform the digestion, we will check once again using electrophoresis, gel electrophoresis. For the restriction enzyme digestions, so basically the DNA here in the PCR products, of course, we will selectively cleave the DNA molecules wherever a short specific sequence is recognized by the enzyme in a process known as restriction target. So in this process, we can use various types of enzyme, but always, this is important, always check the digestion conditions recommended by the restriction company or restriction enzyme manufacturer, because similar enzyme from different manufacturer may need different conditions, so it will work. And here is the restriction enzyme activity examples. So we know 2 types of restriction enzyme activity. First is sticky end digestions, and the second is blunt end digestions. So in sticky end digestions, we notice here that the pattern of the cut is like a zigzag. For example, in this enzyme, ECO R1, so the restriction sites is GAA-TTC. And after we add the ECO R1, the enzyme will cut between G and A. Here, so we can see that the pattern of the cleave is like a zigzag. And we also know blunt end digestions, where the pattern of the digestions is into one straight pattern. So for example, is ECO R5. Here the restriction site is GAT-ATC. But after cut, it will cut between GAT and between ATC. So it means the result will become like one straight cutting lines like this. Another example is SAL1, also sticky end digestions, also XHO1, also sticky end digestions, and also PST1, this is also sticky end, but SMA1 using blend and digestions. This is the final step. After we perform the digestions, we will interpret the result using gel electrophoresis. This is this picture shows one example of the result of the DNA after the digestions. For example, here in the left picture, you can see here it's the band full size 500 base pair. And it's not cut at all. But in the right side here, both of the DNA 500 base pair completely cut into 300 and around 200 base pair here. So it means this is homozygous, this is also homozygous. However, we have also here, we have three bands, the 500 base pair, 300 base pair, and also 200 base pair here. So this is means that we have between two human chromosomes, the one of them is have the polymorphism, so it changed into C, while the other one chromosome still A. That's why we have one band here still 500 base pair, and the other chromosome, because it have the polymorphism, it cut into 300 and 200 base pair here. Another example is here in the right side. The full size is 260 base pair. In the middle, it's not cut, so it means it's still full size 260. But in the right side here, it's completely cut. It means in both the chromosomes, it have the polymorphism, and T changed into C. It becomes the CC here. And in the left side, we can see that it has three bands. It means it's heterozygous, where one of the chromosomes still have the T, but the other chromosome changed into C. That's why it's cut into 184 and 127 here. In this step, I have a tips. So we have to really make sure that we use suitable gel for our electrophoresis. We have to check the ideal DNA size range recommended by the gel manufacturer. And it's better to make gels with similar concentrations and thickness every time we perform the gel electrophoresis. This is just, I want to give you some examples of two products of agarose gel we can use for electrophoresis, agarose S and agarose 21. Here we can see that in agarose S, showed by the red arrow, the nucleic acid fragment separation range between 500 base pair and 30 kilo base pair. While the agarose 21, the fragment separation range between 10 base pair and 100 base pair, 1000 base pair. It means, for example, our DNA product, the band size is only 1000. Of course, it's better if we use agarose 21 compared to if we use the agarose S, because from the specification of the agarose gel itself, it suits agarose 21 better. So this is very important, because if we use wrong agarose gel or wrong gel electrophoresis, it will make harder for us to interpret the results. This is another example of the gel electrophoresis. So here we can see that the full size is 300 base pair, while the cut after the digestions, it will cut into 200 and 100 base pair. It's very clearly that the in lane number 1, it's still 300 base, around 250 base pair, and in line 2, it's cut into 150 and 100. In lane 3, also cut into 150 and 100, while in lane 4, because it's heterozygous, only one chromosome will cut, so we have three bands, 250, 150, and also 100 base pair. This is several situations where we have to be careful when we interpret the result of the PCR reflex. So here we can see that in both picture, the density of the band is very thin. This low density will make it harder for us to interpret the result, because sometimes we are not able to distinguish. For example, here in this picture, the band here, even though it's cut, the density is very low. Sometimes if we are not careful enough, we thought that this is uncut DNA. This is very dangerous. So we have to be very careful when we move from the gene amplification steps into the enzyme digestion steps. Another situation that we have to be very careful is shown in this picture. So in this picture, we can see that here, objectively, we can see that there are three bands here: the upper one, the middle one, and the lower one. Also here, this is also three band, one band, three band. This is three bands, three bands, three bands. But in reality, actually, this is not three bands. The reason of this phenomenon is one, for example, if we use too much DNA, and then the second is if we are not using not enough restriction enzyme. So it resulted that in not all the DNA will be cut by the restriction enzyme. So only partial amount of the DNA will cut. So some of the DNA will cut into two lower band, but some that should be cut, but it's not cut because not enough enzyme. It's still feasible because it's not cut. This is very dangerous also because it may lead us to the wrong genotyping result. So we have to be very careful in this kind of pattern. But in this kind, for example, this phenomenon happens, we can notice by, for example, here in the upper band, we can see that the density is lower than the second band. This is very strange because if it's really heterozygous, it should be the upper part, the upper band should be more denser than the middle and then more denser than the lowest, like in this lane. So the upper band should be denser than the middle and then the lower. But here in the upper one, the density is lower compared to the middle and then the lower. This is one sign that maybe it's not enough restriction enzyme or too much DNA amount we use. And now we are talking about what's the associations of this kind of study, polymorphism, with the gastroduodenal disease pathogenesis. So it will raise question for us whether the gene polymorphism is it a danger. Most of the time, the gene polymorphism is harmless, so it will not give any disadvantage for us. However, in some occasions, the polymorphisms, it means the deviation from normal, may lead to the increased risk to disease or certain health conditions. For example, here, the study by Huang et al. At 2002, the interleukin-1 polymorphisms were related to gastric inflammations and also an atrophy, where we know that gastric inflammation and atrophy are factors thought to be important to gastric carcinogenesis. And another study by Li showed that the interleukin-1 beta T allele is associated with Helicobacter pylori infections in non-cardiac gastric cancer in Chinese populations. Another study we can relate to the polymorphism study is polymorphism in CYP2C19. So CYP2C19 is an enzyme responsible for metabolizing drug in liver. So CYP2C19 polymorphisms may result in different CYP2C19 enzyme activity. So if the mutations not appear, it becomes the rapid metabolizer. If it's appear, it becomes intermediate or poor metabolizer, and also possible ultra rapid metabolizer. And in CYP2C19 itself, we have several axons or several locus. in axon 2 and axon 3 are associated with decreased or diminished activity of the enzyme, while the CYP2C19 in locus 17 is associated with increased activity. So in ultra rapid and rapid metabolizer individual, it makes the enzyme activity may decrease. So because the enzyme activity may decrease, it lead to therapy failure. Therefore, data regarding polymorphism genotyping is also important to consider in daily practice, because if we know the genotyping result, we are able to conduct, for example, like personalized therapy. So we will increase the efficacy of the therapy to certain individuals. Okay, I think that's all. Thank you very much for your attention. Stay safe, stay healthy, and keep moving forward. Thank you.
Okay, thank you very much, Dr. Dohan. It was very interesting presentation, isn't it? For all the participants who want to ask a question, please drop your question in Q&A or chat box. Now, let's continue to the next speaker. The second honorable speaker for today is Professor Ludovic Vallier, MD, PhD, Professor of Biochemistry, MRC from Cambridge Stem Cell Institute, Jeffrey Chia Biomedical Center, University of Cambridge, UK. Before we start, let me introduce Professor Ludovic's profile. Okay, now you can see Professor Ludovic Vallier, MD, PhD CV. He is now working at MRC at Cambridge Stem Cell Institute, Jeffrey Chia Biomedical Center, Biomedical Campus, Pudicombe Way, Cambridge. He has H index for about 55 and total number of publications about 116. The total number of citations is very much. He is very expert in this. So yeah. Now I have the pleasure to invite Professor Ludovic to give the presentation about pluripotent stem cells and organoids to study liver disease. Professor, the time is yours.
Okay, thank you. Hello, everybody, and thank you very much for this kind of invitation to present the work of my lab today. So I'm going to share my screen. Let's see how that works. Okay. Can you see my screen? Is it okay?
Yes, I can see. It's okay.
Yeah, great. Okay, another pointer. Okay, so yes, once again, thank you very much for inviting me and for basically taking the time to listen to the work developed by my lab. Today, I'm going to basically present the work that we are doing on propellant stem cells and organoid to study liver disease. And I will basically show you state-of-the-art data on how we can use those two model system in the context of liver. So just to introduce the liver. So the liver is the biggest organ in the human body, and it's fulfill a wide diversity of function. And so most of these functions are fulfilled by one cell type, which are the hepatocytes. And those hepatocytes are capable to detoxify drugs, have a key metabolic activity for lipids, glycogen, a key storage activity, and also a key production and secretion. They produce albumin, which is the most abundant protein in the plasma. And so hepatocytes represent 90 to 95% of the cells on the liver. So it's really the main factory unit of the liver. The other key cell type in the liver are cholangiocytes, and you will see I will also talk about those cells later on in my talk. And cholangiocytes basically are cells that line up what we call the biliary tree, which is a network of conduits in the liver. You can see that here, that basically collect all the toxin produced by the liver, and that takes all those bad acid and so on in the gastrointestinal tract. So key cells for transport, key cells also for inflammatory response during chronic disease, And also, there is evidence that cholangiocyte could be involved in regeneration of the liver. So those cells during injury of the liver could, in fact, transdifferentiate into a hepatocyte and help to repair the liver after injury. So that's the other point that is important, that those cells in the liver are organized in a very specific architecture, what we call liver lobule, and those liver lobule basically are also extremely important in terms of function, because there is what we call a zonation in those lobules, where the hepatocyte will have different type of function and activity along this zonation. So main message to take home is that the hepatocyte are organized in a complex architecture that are key for their function. So why the liver? So the liver, in fact, is really interesting, because liver disease are one of the rare group of disease that are still increasing year on year, especially in countries like the UK, compared to other group of diseases like cardiovascular or cancer and so on. So that's really one of the rare group of disease where clearly clinical treatment are not available or not working as well as other disease. And that's important, because in UK, US, and other countries, there is currently a pandemic of liver disease associated with obesity and insulin resistance, and basically, which is called non-alcoholic fatty liver disease. So this group of disease basically induced by the accumulation of fat in the liver, which result in a cell death of hepatocytes and induced basically liver failure. And currently, there is no good treatment against this type of disease. And so just to give you a bit more detail, so for example, in the UK, 20 to 30% of the population will have fat in their liver. So vast number of people. And a small fraction of those people will basically progress into disease states, which we call steatosis. And then the steatosis will evolve in a pro-inflammatory situation, where basically the hepatocyte will start to die and induce an inflammatory response, which will basically create a cycle of fibrosis. And then this fibrosis will become cirrhosis, and then the liver is not functional. And most of the time, those liver will progress to hepatocellular carcinoma, liver cancer. And at the moment, there is no treatment to block this progression, or at least there is treatment, but they don't work very well with all the patients. So there is a lack of stratification, there is a lack of information also on prognostic. It's very difficult to plan or to predict how fast the disease is going to progress. And in fact, the only treatment currently available for end-stage NAFLD-NASH is liver transplantation, whole organ transplantation. And that's why basically, our lab is interested to develop, in fact, and study liver disease, because there's a lack of good treatment, and also because we want to basically try to find alternative solution to liver transplantation. And for that, we are basically using human propellant stem cells. And there's two categories of human propellant stem cells that we have in the lab. We have human ES cells that are derived from human embryo at the blastocyst stage, and more precisely from this mass of cells called an inner cell mass, which contain the cell of the embryo proper during the early development. So what we do is that what we used to do is that we take human embryo produced by in vitro fecundation, and then isolate this inner cell mass, put that in culture, and then after a few weeks, we can see sometime colonies appearing that we can grow indefinitely in a dish in vitro, and that likely are what we call human embryonic stem cells, and that are in fact representative of the cells of early development. The other type of propellant stem cells we are using in the lab, which are more and more now replacing human ES cells, are what we call human-induced propellant stem cells. And in this case, those cells are generated by what we call direct reprogramming using 4 transcription factors, OCT4, SOX2, CAL4, and CEMIC, which were first identified by Yamanaka. We got the Nobel Prize for that. And basically, when you overexpress those transcription factors in somatic cells, for example, skin cells or blood cells, what you do is that basically you reprogram the cells into propellant cells that are very similar to human ES cells, and that we call human IPS. And in fact, hence those cells, human ES or human IPS, are extremely similar, and we have never found really a consistent difference between those different cells, or at least those pluripotent cells of different origin. The other model system that I will also introduce today is organoid. And organoid are totally different from pluripotent stem cells. They are generated by growing epithelium, gut, liver, and so on, in 3D conditions in the presence of wind. And this method has been developed by Hans Clever almost 15 years ago now. And that approach allows really to grow in vitro epithelium, adult epithelium. And I will come back later on that in my talk. So let's start with human pluripotent stem cells. So human pluripotent stem cells have two properties that make them uniquely interesting for clinical application. They can self-renew indefinitely in vitro, meaning they can proliferate in the dish. We can maintain them for 20, 30, 50 passages, 1 year, 2 years, and they maintain their unique characteristic of differentiation, meaning that they are pluripotent. They, in fact, can differentiate in all the cell type of the adult body. And over the years, now protocol have been developed to generate hematopoietic stem cells, neural cells, pancreatic cells, endothelial cells from these pluripotent stem cells. And that's really those two properties, this infinite capacity of proliferation and this capacity of differentiation, that really is interesting, because that means that we can now produce an infinite quantity of cells with a clinical interest, for example, disease modeling or cell-based therapy using those cells. That's exactly what we're doing for liver. So what we've done is basically develop a protocol that allows to differentiate human pluripotent stem cells into liver cells in vitro. And for that, what we do is that we follow a natural path of development. Basically, we try to mimic what's occurring during liver development in a dish. That's mean that we have a protocol step by step respecting those key stages. So we start now by the first stage, the endoderm differentiation, which is the earliest progenitors for all the gastrointestinal organs. Then we specify this endoderm into a foregut cells, then this foregut into hepatoblast, which is the earliest progenitor of the liver. And then after we induce differentiation of those hepatoblasts into hepatocyte, also into cholangiocyte. But I'm going mainly to focus on hepatocyte today. And so the cells we generate at the end of this process, which take 35 them in our best protocol, what we observe is that the cells now will express hepatocyte markers like albumin or alpha-1 antitrypsin or HNF4. And they will know this differentiation will be very homogeneous. 90% of the cells, 99% of the cells will be basically hepatocyte. They will have key activity associated with hepatocyte, including ARI cycle, albumin secretion, alpha-1 antitrypsin secretion, but also CYP3A4 activity, which is one of these detoxifying enzyme, which is really key to metabolize drug in the adult liver. So no, that's give you the big principle. In fact, what I'm showing is that this protocol, this method of directed differentiation following a natural path of development is basically used with all the human pluripotent stem cell protocol. If you want to generate a neuron, if you want to generate a pancreatic cells or cardiac cells, you basically apply the same principle, which is try to follow as much as you can a natural path of development with those key steps of differentiation. So what is important is that those cells are interesting and they're useful. You can basically transplant them in an animal model. In this case, it's a mouse model for acute liver failure. So those mice, which is named UPS kids mice, those mice in fact express a transgene that kill liver cells. So they will basically die if you don't transplant hepatocytes. So what we do is that we take those mice, induce the transgene to kill the hepatocyte, and then we transplant to our iPS-derived hepatocyte. And what we observe is that human cells can engraft in the mouse liver, form this very nice colony that expresses human albumin or human alpha-1 antitrypsin, and we can detect human albumin for 8 to 12 weeks in those mice. So that's how the cells that we generate in vitro have the capacity to engraft and colonize the liver, and so meaning that they are functional in this sense. And so what we are also interested in now is to use those hepatocytes to model disease in vitro and to generate new biology. Okay, because what is important is that a number of diseases, especially liver disease, are difficult to model if you don't use a human system. So you have a very good mouse model, but the problem is that, for example, the disease I'm going to show, which is alpha-1 antitrypsin deficiency, the mouse model don't recapitulate entirely the human pathophysiology. And so the goal really now is to use human propellant stem cells to study human-specific mechanism and complement what has been done with other model systems. So I told you, today I'm going to just give you an example with human alpha-1 antitrypsin deficiency. So this disease is induced by a point mutation in the gene SERPIN1A, which code for alpha-1 antitrypsin. And alpha-1 antitrypsin is specifically produced in the liver. And this protein is really important to regulate the activity of neutrophil, especially in the lung. And so when you have this point mutation, SERPIN1A, in the gene SERPIN1A, what happens is that you have a change of configuration of the protein. And the change of configuration of the protein results in an accumulation of polymers. These polymers are going to accumulate in the liver cells, in the hepatocyte, and likely are going to kill the hepatocyte and induce liver fibrosis and cirrhosis. So patients with alpha-1 antitrypsin deficiency systematically end up with a liver transplantation. And so what is important is that when you do the same mutation in the mouse, you don't have liver disease. Okay, so likely we can't study exactly what's happened, why the polymer are formed, why they accumulate, and why they induce cell death. And that's an example of a limitation of a mouse model. And so that's why we decided to use human propellant stem cells, is basically to try to answer some of those questions using a human-specific system. So what we did basically is to take a biopsy from a patient with this disease, with this genetic disease, and we derive human-induced propellant stem cells using those 4 factors of our expression system I've described previously, and that's allowed us to generate human iPS cell line that carry this point mutation that induce alpha-1 antitrypsin deficiency. Then what we did is to correct this point mutation using CRISPR-Cas9 system or using finger, and likely like that we can generate a human-induced propellant stem cell which is corrected. And then after we differentiate the mutant lines and the corrected line into hepatocyte, like that we have a population which is isogenic in terms of genetic background, but likely that have a point mutation versus a wild type genotype. And that's allows us to compare those two situations without any interference of the genetic background. So just to show you that we can indeed model the disease using this approach. So in this case, I show you a hepatocyte generated from those iPS, either with the mutation or corrected or from a healthy donor. And we use a specific antibody to detect the polymers of alpha-1 antitrypsin. And you can see that in the mutant line, we can detect this polymeric form. Whereas in the corrected, of course, we don't see it, as we don't see it in the healthy donor iPS cell line. So that just shows you, very briefly that indeed, we can directly correct the disease and detect the disease in the non-corrected hepatocyte. So then, what we decided to do is to take advantage of this capacity of producing a large quantity of cells from iPS to do study that we can't do with other model systems from primary cells. And we decided to do 2 large-scale analyses using proteomic, by actually trying to compare what is the composition of those polymers, especially the vesicles that contain those polymers in hepatocyte derived from iPS cells, either muted or corrected, and also to do RNA-seq analysis, meaning transcriptomic genome-wide analysis to detect what will be the gene expression profile change induced by the mutation. So just to show you first the protein, the proteomic analysis here, what you can see is that we did a number of comparisons, and we found 17 proteins that are strongly present or absent in the mutant line versus the corrected line. And those proteins, what was interesting first, are a lot of them are involved in cancer, which suggests that again, one of the key consequences of alpha-1 is by promoting transformation of the hepatocyte and hepatocellular carcinoma. But what also we found is that a lot of the proteins were located or related to mitochondrial activity. And that's prompted us to look again into mitochondria in the hepatocyte derived from iPS cells, mutated or corrected. And what we found is that in the mutated cells, we see a lot of abnormal mitochondria with a lot of accumulation and fusion of mitochondria with the ER and with the nuclear membrane. And that's now shown here by electron microscopy, or here using a mito tracker, which is basically an inflorescence marker that stains specifically mitochondria. And that's the quantification of that, showing you that in fact, no, the morphology of the mitochondria in the mutant cells is extremely abnormal. Then, no, that's basically shown that mitochondria are targeted by the disease, and in fact, the polymers really strongly affect the mitochondrial biology, which probably what induce oxidative stress and then induce cell death of the hepatocyte. Then we did the RNA-seq, so basically genome-wide analysis at the transcriptomic level, and we compare again our mutant hepatocyte versus a corrected hepatocyte. And what we found is that there's a signature, transcriptomic signature around 1,000 genes that's distinguish those two populations of cells. And then, to refine a bit more this analysis, because when you do this kind of genome-wide analysis and you get so many targets, of course, it's very difficult to understand which one are important and which one just secondary effect of the disease. And so to have a better kind of identification of gene, what we did is basically to compare proteomic data or RNA-seq data and also RNA-seq coming from primary cells from patients, and try to combine all that in a big bioinformatic analysis. And using this approach, we've been able to identify 20 genes that are basically specifically represent specific signature of disease hepatocyte from induced by alpha-1 antitrypsin deficiency. And what you can see now, that's a few examples of those genes that then were validated into patient samples, so biopsy from patient with this mutation, and comparing wild type biopsy versus patient biopsy. And you can see that some of those genes are extremely strong, for example, collagen 1 and 2, 6L5, L8, pro-inflammatory cytokine, and so on. And so that's mean that by actually using this approach, we have been able to identify biomarkers that are specific for the disease. And one of the best markers we found, in fact, is AKI1B10 and TMP21, which really show basically in patient biopsy evolution of the disease, and basically represent a clear warning that those patients are going to need transplantation very soon. And that's really the two best markers we were able to identify that really systematically basically mark disease progression and can be used as a prognostical almost for disease evolution. So what I've just shown you is that we can use human-induced pluripotent stem cells to basically study new disease mechanism, and that's the key message. So we can basically show that when you have alpha-1 antitrypsin deficiency in the liver, you have accumulation of the polymer in the cells, that we know. That this accumulation of polymers, because they accumulate in the cells, deform the whole ER structures and vesicle transport system, will affect also mitochondria. And those mitochondria basically will result in cell death, and that the cell death and also into induction of pro-inflammatory cytokines in collagen deposition, as I shall show you, and that the combination of both basically is going to result in cirrhosis. And that's really that's the double kind of edge mechanism, where in one case you induce cell death, but in the same time you're going to induce the recruitment of more immune cells. We're going to induce a lot of deposition and change of the architecture of the liver that's going to result into a liver failure. As I said, the key message to take home here is that you can use human-induced pluripotent stem cells derived hepatocyte to study liver disease and to understand new mechanisms and find biomarkers and mechanisms that are relevant for patients. Okay, so now I'm going to switch a bit more into other aspect, and I think it's another important message to take home. So when we use differentiation of human pluripotent stem cells, as I told you, we start from a very early stage of development. And what we know, because we spend a lot of time characterizing those cells, is that all the cells we can generate from human-induced pluripotent stem cells are not equivalent to fully mature adult cells. So when we produce, for example, hepatocyte from human pluripotent stem cells, in fact, they still express fetal markers. So those cells are kind of fetal or less functional adult hepatocyte. And that's important because it decreases a number of functions that could be relevant for, for example, toxicology screen or even disease modeling. And that will be a challenge. And it's true for any cell type generated from human propellant stem cells, cardiomyocyte, neurons, pancreas. They're always like, they are never like fully equivalent to human adult cells. And that's not easy to understand because, as I told you, the protocol we use for hepatocyte is 35 days. But during development, during human life, to generate a fully functional hepatocyte, you basically need 9 months after birth, so basically 21 months. Okay, so basically we try to generate functional cells in 35 days when it takes naturally 21 months to generate a functional cells. And that's so we squeeze a lot of development in vitro. And of course, we think we are missing key step, which result that the cells are not fully functional. So to bypass that, there is different method and different approach that are currently explored by a number of groups. And what we're trying to do in our case is basically to try to grow those cells in 3D. Okay, try to reproduce this architecture of the liver that I've introduced, because we think it's key for function. And so if you can grow those cells in 3D environment, they may acquire more function and become more functional. To do that, we use a system called the raft system, which is basically a solution of collagen where you can mix your cells. And after you aspirate the supernatant, and you get a semi-solid disc where the cells are embedded in 3D. So we differentiate our iPS still in 2D for 23 days, 25 days. We embed them in the raft, and sometimes we add other cell type and co-culture, because we also think that adding additional cells may help with this process of maturation and acquisition of function. So just to show you the results, so that's the raft. Now, as I told you, it's a solution of collagen. We mix our cells, hepatocyte and sometimes other cell type. We aspirate the medium, so we obtain a semi-solid disc of collagen where the cells are embedded in. Okay, and that's what they look like after embedding. So we have big clump cells growing in collagen. So what is interesting that indeed, when we grow those cells in this condition, they acquire, or at least display increased function. So that's, for example, albumin secretion. And you can see that's now the 2D conditions, right? That's a 3D condition. And you can see that we secrete much more albumin in those 3D conditions. The same with alpha-1 antitrypsin, a bit more. But what also is interesting is alpha-fetoprotein, AFP, which is a key fetal marker. So AFP, for example, is expressed for a long time in fetal hepatocytes. And it's marked by maturation of the liver. After birth, your AFP decrease and disappear on 9 months. And that's show that you have reached full adult maturation of your hepatocyte. So when we see this marker in all culture conditions, that means our cells are fetal. And when we see disappearing, that means that they are progressing into maturation. So that's what is interesting, that in 3D, clearly AFP drop, meaning that the cells are a step further in terms of adult function. And if we do again gene expression profiling, what we see is that all cells grown for 45 days in 3D are much closer to adult hepatocyte. They're still not identical, okay, so that's still a limitation, but they clearly better representation of adult liver cells. And that's also shown by different aspects. So, for example, if you look here, MRP2, which is a protein that is really important in the liver because it's marked what we call caniculae, which are small conduits between hepatocytes that collect all the bile acid, all the toxin, and that drive that to the biliary system. And basically, when you look at in 2D, you don't have a lot of organization or expression of MRP2. When we are in 3D, we have those nice caniculae formation, suggesting that again, you have now creation of a network that is functionally here basically, but it's not the case in 2D. The other interesting aspect is that basically when we grow those cells in 3D, we can keep them much longer in culture. So in 2D, after 45 days, they will start to die, whereas in 3D, we can grow them for more than 100 days and they will be fine. And that's means that we can do chronic treatments, what we can do with 2D. So, and that's also translates into additional function. So for example, to model a non-alcoholic fatty liver disease, what we do now in the lab is to feed the cells with fatty acid, okay, which basically is the source of lipid that induce naturally through our diet. And here, when we put oleic acid on the cells, for example, that's a key fatty acid in our diet that's accumulated in the liver, we don't see lipid droplet in 2D derived iPS hepatocyte. Okay, so there's no lipid accumulation. Whereas when we go in 3D in a RAF system, what we see is that we have this beautiful droplet of lipid accumulating very quickly, 24 hours or 7 days. So that means that in 2D, the cells are not able to accumulate lipid very well, whereas in 3D they are, and that's probably just because of this 3D organization. And what also is interesting is that if you take palmic acid, which is another fatty acid, which is known to induce cell death in hepatocyte, we know if we put palmic acid on 2D cells, they never die. Whereas when we're doing that in 2D, in 3D, they start to die very quickly after 24 hours. And that's shown here. If you look at cell viability, say, you see that the cells after 1 week and even better after 4 weeks are dying very quickly. So what I'm trying to show is that now using this 3D approach, not only we improve the function of the cells, but we can also model disease or mechanism that we couldn't in 2D. That's also true for detoxification. So that's in this case, paracetamol injury. So paracetamol overdose is one of the most common cause, or at least one of the most important cause of urgent liver transplantation. It's basically teenage kids that decide to kill themselves by absorbing paracetamol, and the paracetamol will basically, when it's ingested in too high dose, will induce the production of a toxin, this toxin here, N-acetylbenzoquinone, which basically will accumulate in the liver and will kill hepatocyte very quickly and will induce acute liver failure. And so, basically, if you put the paracetamol on cells in 2D, you don't see cell death, because simply the cells will process paracetamol, but we secrete that in the medium and we change the culture medium every 2 days. So basically, we won't see this accumulation, this toxic, the accumulation of this toxin. Whereas when we go in 3D, then we see this cell death, and that's shown here. For example, where you have increasing dose of paracetamol, and that's basically the quantity of cell death in the dish. And you can see that when you increase dose of paracetamol, you see an increase of this cell death. What is important is that this cell death is specific for paracetamol injury, because if you use N-acetylcysteine, which is a drug that you can use to stop the overdose of paracetamol in patient, what you see that you block this spike here. Cell death. And finally, the other point is that we can add other cell types like macrophages, which are known to be part of the pro-inflammatory feedback loop that induce acute liver failure, and we can observe an increase in cell death using these cells. So what I'm trying to show you here is just again, with this 3D system, we can model detoxification mechanism and also a disease like acute liver failure induced by paracetamol overdose. And finally, the last point I would like to show you is that now we can also add a lot of different cell types that are important in the liver to try to see if we can reproduce this architecture and increase function. So that's what I would call the Rolls Royce of a model system, is when we mix the hepatocytes, cholangiocytes, but also stellate cells and macrophages, so two other cell types, and Kupffer cell, two other cell types of the liver. And in this case, the hepatocytes express the GFP, the cholangiocytes express the red FP, the LX2 are in blue, and macrophages are basically in yellow. And what you can see is that the hepatocyte-like cells will organize around the cholangiocytes. So basically, the cholangiocytes will form tube like they will do in vivo in the liver, and then the hepatocyte and the LX2 cells and the macrophages will start to self-organize around those structures. So it means that when we mix those cells together, there seems to occur some architecture that mimic what is happening in vivo in the liver. And what is interesting is that in this case, we also gain a bit more function. We have a better expression of albumin, a lower expression of AFP. CPA4 doesn't increase very much, but that's still a challenge. But what we also see is pollution of bile acid and transport of this bile acid by the cholangiocyte. So that just shows you that we can even complexify further the model system, and that now we can produce all those cell types from different sources, mix them together, and try to have a self-organizing mini organ in vitro. Okay, so that's what we biologically, that's probably the most complex model system we can generate right now with IPS. It's 4 different cell types. It's long-term culture, is 3D. So you can't do much more complicated than that right now. And clearly, those model systems now allow us to model complex disorders and a wide diversity of disease and in a dish. And as I told you, we're starting to biologically generate new biology and understand and discover new targets for drug development. And we hope also develop new treatments using those cells. So now I'm going to switch to the other system we're using, which are organoids. And so as I told you at the beginning of my talk, organoids are derived from epithelium, adult epithelium most of the time. And basically, in the liver, what we do is that we derive organoids from the biliary tree. Okay, the only place where we can derive organoids is the biliary epithelium. And so what we do is that we take different pieces of this biliary tree, put them in culture in 3D, in matrigel, which is basically the matrix we use for this 3D culture, in presence or an absence of wind. And now they form this organoid that we can then grow in vitro most infinitely. That's now show you a bit more detail of we do that. So that's a gallbladder from a patient that underwent transplantation. We take this gallbladder, we scrap the cells that cover this gallbladder, which is part of the biliary tree, and that's the cells of the scrapping. And then we put them in culture in presence of aspondin, matrigel, and what we obtain is those spheroids that we call, in fact, cholangiocyte organoids. In this case, they derive from the gallbladder, which is outside the liver. We call them extrahepatic cholangiocyte organoids. And no, you can do with that from different regions in the biliary tree, from the intrahepatic biliary tree, so the part of the biliary tree which is in the liver. You can do that from the gallbladder, from the common bile duct, which is basically the tube that links the liver to the digestive tract. And you can even do that from the pancreatic duct. And each time you will generate those spheroids. And so that's just, that's very efficient. And we've been deriving those cholangiocyte organoids from a broad diversity of patients. And we don't see any differences in terms of efficacy of defense of derivation, and gender or even age. No, it's just very efficient. And so those cells that we derive in vitro as organoid can be grown for a prolonged period of time. You can grow them for 20, 30, 40 passages. They maintain normal karyotype, and they will maintain expression of key markers for biliary cells, cholangiocytes, including CK19. So that's the morphology. They will have no CA, which is a specificity of the cells. They will closely resemble primary cells. And what is interesting that they will express key markers, but they will express those key markers in a polarized fashion. For example, in this case, somatostatin receptor is expressed at the surface of the organoid, whereas ASBT is expressed in the lumen. And this polarization is really important for the function of the cholangiocyte, meaning that they are basically functional cells in vitro. And we can even further demonstrate that by showing that those cholangiocyte organoids can transfer bile acid, so that's a fluorescent bile acid, that they react to hormonal stimuli, for example, to secretin and somatostatin. They will swell basically. They'll express ALP, alkaline phosphatase, and that they will secrete gamma GT, which is a key marker secreted by cholangiocyte. And the expression of those cells in vitro will be equivalent to the secretion that you will observe by cells in vivo in an adult patient. So that just means that now we can grow those cholangiocytes in vitro from different regions of the liver, and that they form those organoids, and they are basically very similar to primary cells. And so now we're trying to think of how we can use those cells. So of course we can use for this modeling, that's what we're doing, but we're also interested to generate new therapy for liver disease. And in this case, one of the key liver disease is what we call biliary atresia, which basically affect the common bile duct. So it's basically kids that are born without the common bile duct, and so their liver is not properly connected to the digestive tract. And of course, these kids very quickly end up in trouble, so they have to undergo a surgical procedure called a Kasai procedure, which bind their liver to the gut, but sooner or later they will need a liver transplantation. So to basically try to address this clinical need, what we decided to do is to see if we could build an artificial common bile duct in vitro using cholangiocyte organoid derived with the method I just showed you. To do that, what we do is that basically the idea we had is to basically develop an artificial common bile duct and test that in the mouse first. Okay, and it's very simple. We develop a model where we basically cut the common bile duct in the mouse and then replace it by an artificial common bile duct made with human cells. And I'm just going to show you a few data on that. So to build this common bile duct, this tube that links the liver to the gut, what we do is that we basically develop a collagen scaffold. So we mold a scaffold using this approach here. It's basically, a tube that we create with collagen. And we obtain this kind of construct here that we get basically cut. And then we simply put cells, cholangiocyte organoids derived from patient, grown in vitro. We put those cells in the scaffold, grow that for a few weeks in a bioincubator. And where then at the end is basically a scaffold which have been colonized by those cholangiocyte organoid. And they express the usual biliary marker, CK19, CK7, for example. So you here with GFP expressing cholangiocyte. And that's a reconstruct collagen bile duct basically. And then what we did is take this, take out the common bile duct in the mouse. So it's very tiny, less than a few millimeters in size. So that's basically, you need very good surgeon to do that. And our collaborator, Squash Parsi, is excellent in doing this kind of microsurgery. So basically, you cut the common bile duct of the mouse and you bring your human collagen your collagen human cholangiocyte bile duct here, and you suture this bile duct to the liver and to the digestive tract. Okay, and we did that with cholangiocyte, but we also did that with fibroblasts as a control. And what you can see is that after a few weeks, we still have a nice clean construct. After transplantation, after a few weeks, but when we use fibroblasts, we have a lot of fibrosis, and basically the mice die in a few days. And whereas with the collagen scaffold reconstituted with human cholangiocyte organoid, we have a nice lumen, and we have still transport and circulation in this reconstituted tube. And now, if you do histology, you can see that the cholangiocyte are still here. So that's human cholangiocyte that will express the usual CK19. They have this nice morphology, which is typical from a common bile duct epithelium. And also, if we do MRPC, we can see that we have a nice and functional common bile duct in those mice when you use the human cholangiocyte. And of course, finally, what we shown also is that when we reconstruct those common bile duct, we can rescue the mice, because the mice with common bile duct will die in 24 hours. And so here, we can show that basically, when we use those human cholangiocyte reconstructed bile duct, we can basically rescue the mice and allow their survival for several months with a problem. So that just shows you that we can use those human cholangiocyte to basically reconstruct common bile duct. And currently we're developing a new version of this system, of course, to have a human-sized common bile duct that we could use in the context of biliary atresia and other diseases affecting the common bile duct. So just a small pause here is also, as I told you at the beginning of my talks, that cholangiocytes are involved also in regeneration and can transdifferentiate to hepatocyte. And that's been a long discussion in the field and a key important question. And so we wanted to check if the cholangiocyte organoid that we grow in vitro have this capacity to transdifferentiate into hepatocyte, because of course that could be super interesting to have another source in vitro to produce hepatocyte, again for cell-based therapy and potentially for disease modeling. So what we did is that we took a cholangiocyte organoid from all the different regions I mentioned, common bile duct, gallbladder, intrahepatic, and pancreatic duct, and then try to differentiate those cells in vitro into hepatocyte. And the only message to take home is that indeed, cholangiocytes derived from intrahepatic duct can start to express hepatocyte markers, but it's very inefficient and it's very low. That's just to show you here that the expression of albumin, comparing expression of albumin in those intrahepatic cholangiocyte organoid versus primary hepatocyte. So that's mean that basically only cholangiocytes inside the liver have a very low capacity to transdifferentiate into hepatocyte. So we're still exploring that to see if we can improve further this system. So what I've tried to show you is that really the cholangiocyte system is a very basically efficient method to grow adult cholangiocytes in the dish. Okay, so those organoids are really nice because you really can grow them in vitro, and they really represent cells of the adult. So it's very different perspectives on human iPS-derived cells. Those human cholangiocyte grown as organoids can be used immediately for cell-based therapy because they are already the cells you want to grow. Okay, so it's a promising system. The only limitation, of course, is that those cells will only work for the BRI system. We can't derive hepatocyte organoids yet, so that means that we can only grow cholangiocyte. And of course, those cholangiocyte organoids are limited to diseases that affect the biliary tree, which a small fraction of the diseases that affect the liver. So, for example, we can't choose cholangiocyte organoid to treat NAFLD or non-alcoholic fatty liver disease. And that's clearly going to be a challenge to develop new methods that allow us to grow hepatocyte organoid. And also, what is important that there's still a gap between the culture condition we have developed in the lab, because we're growing that in matrigel, in 3D, in very complex culture system, which is not compatible for clinical use. And we still have a lot of work to transfer those conditions into a GMP clinical environment. Okay, so just to conclude my talk, what I would like to say is that basically, human propellant stem cells can be differentiated into a diversity of liver cells. Now, I show you hepatocyte. We can do also cholangiocytes, stellate cells, macrophages. We can all grow them in 3D culture, which improve their maturation. It's still not the magic one that will allow you to generate fully mature hepatocyte, but still, it's a better system to model a certain number of diseases, as I showed you, detoxification for paracetamol overdose, but also for NAFLD and other complex disorders. What is important is that now we know well those cells, and despite those limitations of functional maturation, we can already use them to basically discover new biology, new mechanism of disease, and by combining data from patients in this in vitro system, we'll be able to develop to identify new target for drug development, but also new marker for prognostic and diagnostic. This model system and the organ system are complementary. In one case, we have cells that follow a natural process of development, so that's going to give us a lot of information on the impact of disease during fetal life. And the other, you have this adult system, the organ system, that really allows us to look at disease in the adult context. And basically, they can be all complementary. Both systems can be used for cell-based therapy. And basically, that's probably the key message to take home is that each cell type, each model system, iPS, organoid, have drawbacks and advantages. And basically, that will mean that those cells will not be used for all the application, but can be, knowing their characteristic, used for specific application. And that using this panel of model system now, we can really look at different disease, different mechanism, and different cell-based therapy or modeling. Okay. To conclude my talk, I just would like to thank the people that have done all the work in the lab. So all the work I've shown in cholangiocyte was mainly led by Fotis Sampagetis, which created the cholangiocyte organoid system. Very talented clinician that is now having his own group. The work on 3D culture conditions was developed first by Trey Geisek and now by Carola Morel, which is also a talented postdoc in the lab. The work on alpha-1 antitrypsin deficiency was generated by Kara Segaris when she was in the lab. And all this work was done with the support of the ERC, which is our main source of funding in the lab. And I would like to thank you for attention. I would be happy to answer any question later in the seminar. Thank you very much for your time.
Okay, thank you very much, Professor Rodolphic. It was very interesting presentation. Now we come to the discussion session. To all the participants, if you have any questions, feel free to raise the question on the chat box. First of all, we can read the questions from the participant. I guess this question is for Dr. Dohan. Okay, Dr. Dohan, are you ready? The first
Yes.
question, okay. The first question is, what is the most difficulties of technical problem during polymorphism experiments?
Okay, thank you very much for the questions. So basically, for the PCR reflip itself, the experiments or the technical thing, it's not too complicated actually, because all the technique is very basic techniques. However, we have, like I said, we just have to be very careful in the each steps, and then for the pitfall, the situations I explained in the presentations, we have to be very careful about how to interpret the result. So, but basically, for the difficulties itself, I think it's not so difficult, because basically this is just a basic skill lab actually. Thank you very much.
Okay, thank you. The second question, it was mentioned that PCR RFLP weakness is that it cannot be used to detect many kinds of polymorphism. What kind of gene polymorphism that can be identified with PCR RFLP method? Can it be used for HLA polymorphism detection? Thank you.
Okay, thank you very much for the questions. So the question is it able to be used to check the HLA polymorphism? Okay, so basically, this method itself we can use to check the polymorphism in every genes. What I mean by it cannot be applied to what I mean in the presentation is we cannot use it to check the polymorphism simultaneously in 2 or 3 or 4 genes immediately. So because we have to check it one by one using this method. So, but in the application itself, we can use this method to check the polymorphisms in every genes, I think. And for the application for the SLA polymorphisms, I think I checked in the PubMed. I think previously there are several studies already checked the HLA polymorphism using PCR RFLP. So it's possible, and if you need some reference, you can check it in the PubMed. Thank you very much.
Okay, that's the answer for the question. Now, the next question is for Professor Valier. For Professor Ludovic Valier, there's a question. Is there a limit of length for CBD replacement using the artificial graft due to CBD vascular distribution?
Yeah, that's a very important question, because in human, the common bile duct is relatively long, and you have vascularization, which is quite important, because no, if you have ischemia in this common bile duct, the cells will die. So we currently doing some work in large animal model, pigs, where we basically we have a common bile duct, human size, and what we see is basically when we transplant this common bile duct, you have recolonization by endothelial cells and vasculature. So we become vascularized very quickly. So we don't think it's a major challenge, because basically, if you do the right transplantation with the right scaffold, basically you will have vascularization of the construct, and you won't have ischemia. But it's a really important point.
Okay, thank you very much for your answer, Professor. The next questions, we have the questions for Dr. Dohan. Okay, the question is, after the PCR, the band density was still low. What should we do to overcome the problem? Because you said earlier, I guess, that before to obtain the best result, we have to get enough density, isn't it?
Sorry, I cannot hear clearly the question.
The question is, if after the PCR, the band density was still low, what should we do to overcome that problem?
Okay, so this is very technical. Okay,
Yes.
for example, the density of the band is not satisfying. It means actually the protocol itself it succeed. I mean we amplify the DNA, but it's not sufficient. So another option is we can redo the experiment of the PCR, and then we can use higher amount of template of the DNA, so we can get higher amount of DNA also possible. Another option is we can perform another PCR, and then we can concentrate. I mean, like we can purify the DNA and then dilute it in lower amount of dilute, so the concentration is higher, like that. So basically, the safest way if I have to do it, I just redo the PCR method.
Okay, thank you very much. The next question from Professor Ludovic Valer. HPSCs can develop into several liver cells. How can we specify to develop the HPSC cell to only our interested cell?
Yeah, so that's a very good question. So the way we do things now, it's basically we take knowledge from human development, I mean, or from development biologists basically, that have studies that have made a lot of investigation on the mechanism that allow the generation of specific cells. So for example, for hepatocyte and cholangiocyte, developmental biologists have shown that they come from a common progenitors called the hepatoblast, and that is this hepatoblasts can differentiate either in cholangiocyte or in hepatocyte with a combination of Wnt signaling, TGF-beta signaling, and not signaling. What we do is that we try to basically do the same thing in vitro, and of course, the key challenge is to find the right concentration, the right timing of those different growth factors. So we have spent a lot of time now screening different culture conditions, different dose of those growth factors to basically be able to generate those different cell types. And so now the key message that we follow principle of established by development biology. So we need a lot of basic knowledge. And then now we adapt that to in vitro conditions that of course are a simplification of the human embryo or the fetal development. But nonetheless, we're trying to respect those big principles.
Thank you very much. The next question for Dr. Dohan. There's a question. Is there another method to obtain polymorphism detection result besides using PCR RFLP? Can it be obtained by using common PCR?
Okay, thank you very much for the questions. So actually, for the methods we use to detect the polymorphism, it also depends on what kind of polymorphism we are going to check. Like I said previously, that there are several types of polymorphism. For example, like SNP, just one of them. Another type is like the short tandem repeat. So for short random repeats or variable number tandem repeats, we can check using PCR, and then we just check the length, because in short tandem repeat, there are multiple repeats of sequence, so it makes the length of the product is different. So in that case, we can just use the common PCR, normal PCR. But in case of FNP, we can, other than refleet, we can also use PCR followed by sequencing. We can also use hybridization methods. We can also analyze the data using NGS data. So actually, in reality, we have various kind of methods we can use.
Okay, thank you for your answer. The next question for Professor Ludovic again. Thank you for the insightful presentation, Professor Ludovic Valer. As we know that liver organoid systems are suited for translational research and drug development and may find clinical application for regenerative medicine in the near future. What is the most challenge and difficult to apply it? Hello?
Yeah, again, that's a very good question. So first, I think the first answer to that is to really we need to really understand what the organoid cells we go in vitro. Okay, it's sometimes a bit oversimplified, to think that those cells are strictly similar to adult cells. Not actually true. For example, we know that when we take cholangiocytes out of the biliary tree, they lose their original identity. So cholangiocytes now localized in this different region of the biliary tree have different transcriptomic profile that they lose when you grow them in vitro. So that's means that we need to really understand that, because that's of course help us to know how we can use those cells. And the field have been a bit sometimes simplistic on looking at those cells. The other, so that's no more conceptual aspect. At the practical level, it's clear that for cell-based therapy, the key challenges are associated with the method that we use to grow those cells. So those cells are grown in matrigel, okay, in 3D. And so those culture conditions are not compatible with very large-scale culture, because it's fairly complex to establish. And also the matrigel is not compatible with GMP production. So basically the cells we have in the lab are a bit difficult to transfer into the clinic. And so that's why right now people are working on a lot of culture system using artificial hydrogel that could replace matrigel and allow really basically to generate organoid in GMP conditions, that are conditions that are required to go for clinical trial in man. And that's something that we're spending a lot of time. And there's been good progress. Now there is extracellular matrix, artificial, that can be used to grow those cells. And it's ongoing, but clearly there's still those challenges, those limitations that are more kind of very linked with the cell culture aspect, really the practical aspect.
Oh, okay. Thank you very much for your good answer. There's another question for you. It seems that pluripotent models for liver disease could restart the hepatocyte from stem cells model. Is it any concern of applying this model for hepatitis C infection? As we know that HCV could reside to our body in dormant state, and it could relapse and infect when there is an available hepatocyte.
So we're talking about infectious disease, or
is it like it seems that pluripotent models for liver disease could like replace the hepatocyte from stem cells model. So is there any concern of applying this model for hepatitis C infection using the hepatocyte stem cells to what can I say, to for hepatitis C infection treatment?
Okay. I mean, so we know that the cells we generate in vitro, the iPS hepatocyte, can be used to. I'm not sure I'm answering the question properly, but I'm going to try. So that we generate in vitro can be used for infectious disease. Okay. So to model infectious disease, for example, we use those cells for. Okay, so we use this model for malaria, for example, because malaria is one of the diseases that go to the liver. You can model Hep C with those cells. It's a bit more complex because you need those 3D structures to get the Hep C. So it's a complicated model to use with the virus, but that's been shown to work. And basically, we know that we can model that. The second part of the question is that so, and of course, so that's for modeling infectious disease. In terms of using cell-based therapy for patients with Hep C, it's clear that it's not a great solution, because it's known that when you do liver transplantation for patient with Hep C, the liver will get contaminated, and basically the patient have redeveloped the disease. So but what we think we could do, for example, is to generate hepatocytes that don't express the receptor of the virus and could be resistant to infection. And that's something that we are looking into, because basically with the genome editing method, the CRISPR-Cas9, we can develop hepatocytes that have increased function or decrease sensitivity to infection. And that's something we're looking into in the lab, actually. I hope I've answered the question, but if please ask me again if you think that's it's not the right answer.
Okay, thank you very much. And then the next question for you, is there any complication after the artificial bile duct transplantation? Like any possibility of complication?
No, so far, large animal experiment, we don't see any complication. I think it works well. Basically, we don't have any occlusion or or no fibrosis or this kind of process in the construct. No,
So
I
this
think
is
yeah.
so there is there's no further clinical trial for the methods.
So no, we no, so we showed that in the mouse. Now we are in a large animal model, and then the next step is to go to human system. So that's something we no we're working on, but no, we are developing the GMP conditions and so on. So it's a much more complex. So no, we process because we need to go to regulatory agency and so on. So that's really where we're going now. We're really going to human. So we have proof of principle in animal model, small and large, and now we're going to human. And we don't. So the big challenge is just now is to solve the technical issue associated with GMP culture, and then go to the regulatory process, which can be fairly complex for this kind of really first-in-man clinical trial.
So it is very promising, I think. Very
Yeah, no,
promising.
I mean, yeah, the data in animal model have been incredibly convincing and really really efficient.
Okay, that's very good. It's very interesting and very impressive presentation from Professor Ludovic. So I guess like we all know reaching the end of the session of discussion session. We would like we would like to capture our screen as the documentation of this workshop. So maybe for all the speakers, please turn on your camera. Can turn on the camera. We are coming to the photo section because at the end, maybe
On the camera.
Professor Ludovic, do you mind to open your camera? We are taking a picture together to documentation. Thank you very much. Okay. Okay, thank you. It's finished for the photo. Thank you very much. Thank you for all the speakers, Dr. Dohan and Professor Ludovic, for the insightful sharing, and to all the participants who actively participated in this section. Thank you also for Dr. Blaise Assoumani for joining us today. To all the participants, we would like to take a moment completing our satisfaction survey we sent to the chat box on this workshop. We encourage all the participants to fill the questionnaire, and then the certificate will be sent to your email. Once again, thank you for joining our workshop today. We do hope you always in good health and wishing you good luck for the future. Stay healthy and stay safe for everyone. My name is Rizky Amalia. I'm sorry for any inconvenience may occur during the workshop. We hope you had a great time with us. Don't forget to join us tomorrow in the second day. Tomorrow's topic is gastric microbiota from sampling to obtain the sequence by Takashi Matsumoto, PhD, and tools and approaches for analyzing microbiome by Dr. Langgeng Agung Resito, MD, PhD. Okay, see you tomorrow. Thank you very much for all.